So welcome back friends. In this video tutorial we will going to talk about the Ames test. This will be the third video lecture. In previous two video lecture we have talked about the Mesensal and Stahl experiment, and the second one was the Hershey and Chase experiment. Those two was from the molecular biology background and this one is from the genetics background. So let's discuss. So it was developed by Bruce Ames, and according to the name of the Bruce Ames, the test was Bruce Ames test. Okay, and Bruce Ames developed this test at UC Berkeley. Ames test is also known as this test is used to determine the mutagenic potential of a chemical in animals. Okay, so what does it mean? This simply means that Ames test can tell us whether a chemical can cause mutation in our body or not. Okay, so this test is based on the capacity of a chemical to revert the mutation in the histidine operon of the Salmonella typhimurium. Okay, so this kind of Salmonella typhimurium is not a normal kind. We are taking a auxotropic strain of Salmonella typhimurium. Okay, and this kind of strain is also known as tester strain. Now the question is, what do you mean by the auxotropic mutants? So auxotropic mutants or auxotropic strain simply means this kind of bacteria should lack the ability to produce some kind of essential nutrients. Okay, so here the auxotropic mutants of the Salmonella typhimurium cannot produce histidine. Okay, and this may be, not maybe, this is due to the frameshift mutation on the histidine operon. Okay, so it is necessary to remember about the name of the mutation that is going on here, and it is frameshift mutation of the histidine operon. Okay, so the idea is we will culture Salmonella typhi histidine negative bacteria in a nutrient media. Okay, and this nutrient media will not contain any source that can provide histidine. Okay, and then we will add chemicals at the middle of that plate. Okay, and then we will incubate them at 37 degrees centigrade for 24 hours. Okay, and after 24 hours of incubation, if we found that colony has developed uniformly, then we can conclude that yes, the chemical is mutagen, because this chemical have the ability to revert the mutation on the Salmonella typhi. Okay, but if we do not get any kind of colonies, or we get some random colonies but they are very small in numbers, then we can conclude that the result is negative, the chemical is not a mutagen. Okay, so let's see how the test is conducted. So we will streak bacterial culture in a petri plate, then we will add the chemical at the middle of this plate. Okay, and this nutrient media will not contain any kind of source that can provide histidine. Okay, and also this media contains rat liver extract. Now the question is, why rat liver extract? Because there are various type of chemicals that are initially in inactive form, but they become activated when some liver enzyme acting on them. Okay, and as a result, those kind of chemicals usually are non-mutagenic, but when they enters inside our liver, they become mutagenic. Okay, so those kind of components or those kind of molecules cannot be detected until we provide the same environment that our liver provides. Okay, now we cannot use human liver for that purpose, right? It is unethical, and that is why instead of human liver we are using rat liver extract. Okay, that will create the condition where this kind of chemicals can become activated. Okay, So that is the concept why we are using rat liver extract. Okay, then we will incubate them for at least 2 days at 37 degrees centigrade. So I have just told that 24 hours, not it is 24 hours, it will be 48 hours, sorry for that. Then after incubation we will get 3 possible results. Okay, so if we look at this result, so the chemical can diffuse, right? And that is the reason why we will get colonies uniformly. That simply means that this kind of chemicals is a mutagen. Okay, and you will not get any kind of colonies just here, because those kind of chemicals cannot those kind of chemicals will not allow growth of any colonies here. Okay, so if you get number of multiple number of colonies in the petri plate, then you can conclude that the chemical is mutagen. Okay, but if we do not get any kind of colonies, then you can conclude that your test is negative. Okay, although you will not get any kind of situation here in reality, instead you will get this kind of situation, that is the possibility number 3, that in that possibility you will get some colonies randomly, okay, not uniformly. Okay, and this is due to the spontaneous reversion, because bacteria also have the ability to undergo spontaneous mutation. Okay, that is why there can be a possibility you will get some colonies randomly, and this is considered as false result, and this is solely due to spontaneous reversion. Okay, now there is a interesting MCQ that I want to discuss, that they can also ask you that except Salmonella typhimurium, what type of strain you can also use. Okay, so we can also use E. coli WP 2UVRA strain. Okay, this is important to remember. They can also ask you this type of questions. Okay, so that's it. That is all about the Ames test. So here we are first streaking bacterial cultures, that is the oxotopic strain of the Salmonella typhimurium. Then we add chemicals at the middle of this plate. Then we incubate them for 2 days at 37 degrees centigrade. Then upon incubation we will get 3 possible results. Okay, if we get multiple number of colonies uniformly, we can conclude that yes, our test result is positive, that means our chemical is a mutagen. If we will not get any kind of colonies, then we can simply conclude that it is a negative result, that means our chemical is not a mutagen. But if we get some colonies randomly, then we can conclude that your result is false result, and that is due to the spontaneous reversion, okay, caused by the bacteria itself. So that is the test that help us to determine the ability of a chemicals to cause mutation in the animals. So I hope this video will be helpful. Thank you.